All right, today we're going to continue our DNA talk, and we're going to look at DNA replication. The word replicate. Replicate means to make more, make a copy of. And so that's what we're going to look at, how DNA makes more DNA, how DNA makes copies of itself. So first thing we look at here is all eukaryotes, and we're just talking about cells with nuclei, have DNA. All right? DNA replication takes place in the nucleus. You want to keep that DNA inside that nucleus because it's safe there. All right? If you have $1 million, you don't just leave it laying on the porch. You lock it up in a safe. Well, that's what we have here. DNA is that important, to us, to living organisms, to the cell. Okay. The process that the cell uses to make new DNA is called semi-conservative. So that prefix semi here, semi means kind of, sort of, only used in part. All right? And that's what happens when we look at our DNA strands. So here's our DNA, original strand of DNA, double-stranded, two sides, got that ladder. We have, and we'll go through this process, a split. We split that DNA in half, and we build two new halves here. All right, so we build one on this side, we build another one on this side. That's going to take that one strand, one double strand, turn it into two double strand. But each of the new ones, so we have two new copies of DNA, each one of them has a piece of old, all right, has a piece of old DNA, and a freshly made piece of new. That's what semi-conservative means. All right, we're conserving half of it and building half of it. All right, so not all brand new, but we have two new strands that are exactly the same now. And part of it was already there, part of it is new built. All right, so that's semi-conservative. All right, the process. Okay, so that DNA strand, normally it's twisted all up in that double helix. Well, it gets untwisted, laid out flat like that ladder, and then we have an enzyme. Remember, enzymes, they make reactions happen. That enzyme is DNA helicase. All right. So on the diagram, you see it right here, the little red ball. That helicase goes through and it splits that DNA open. It breaks those hydrogen bonds, and that's what we have to have. We have to have those hydrogen bonds broken. This thing will keep running all the way up the strand, and it'll break those hydrogen bonds so that we can split those two strands in half and build new. All right. Where this is occurring, at that exact point where it's happening, is called the replication fork, because it looks like a fork. You got a fork with the tongs and the little split. That's what's happening. So we call it the replication fork. It's the spot to where replication is happening at that moment. Okay. All right. Once that strand is split, here's our helicase right here in the middle. We have those double strands split in half. Then we have another enzyme called DNA polymerase that comes in and builds new. So if we have two strands, two halves, we have to have two these polymerase molecules come in and start building. And so they attach to it, one attached here, and it starts building. It starts adding all the way down. And it's going to keep adding. And what's it add? It adds more nucleotides. Get the bell again today. And so we have A's, T's, G's, C's, all different. All right, so it's adding those bases as it goes, matching them up, A's with T's, C's with G's. So where do these bases come from? Well, inside the nucleus, there's kind of like a scrapyard, a junkyard that has all of these different bases. It's got millions of A's and G's and C's and T's all in there. We call those free nucleotides. All right, they're just floating around in the nucleus, and so this polymerase grabs them, pulls what it needs, plugs it in, pulls what it needs, plugs it in. Have to have all that. You got to have protein to make those. So there we go. You got to have that in your diet to build all this stuff. Okay, and so one of these polymerase is going to work one direction. The other polymerase started at this end and is working this direction. They work opposite of each other. Why? it's a little bit more in depth than what we're going to get into. That's more like AP Bio, but you think about it is you always want things working opposite each other so they don't crash into each other. I mean, that's an easy way to remember it. Okay. So to go back through the entire thing real quick, just another reminder. So we started with this one strand, double-strand DNA. We had helicase come in the replication fork, split it in half. We have DNA polymerase on both strands come in, build the new complementary strand. And now, once that happens, the double helix reforms. It all twists back up. That's the last step. And we have two new semi-conservative daughter DNA strands. All right. This cell now has two copies of DNA in it. Why do we need that? Well, we're going to get to that when we get to the next part with the cell cycle, because we have to make new cells. Our cells don't last all the time. They're not going to last forever. If you brush your arm right now, take your hand and brush down your arm, you knock off about 500,000 skin cells. Wow. That's a lot of cells. Have to replenish, okay? Have to build new. And so that's what the body's doing. These cells are doing is they're making new copies of DNA. So we got one DNA, we make two. Now that cell can split in half. We got two cells from one. All right, we're going to talk about that split in half and making new cells in the next set of notes. All right. That is all for the day. Have a good one.